Welcome to a video to describe complement. Complement is a set of proteins that are produced by the second line of defense in an attempt to assist in coordination of activities. Complement is going to function in part to help out with the second line of defense. However, also will occasionally assist the third line of defense. And as such, the name complement is designed to reflect the property of complementing or assisting these lines of defense. So let's get started. To consider complement, a few things that we ought to be familiar with. Complement is a series of 30 different proteins. The function of this video will be to look at a handful of those proteins. These proteins function to support the second line of defense in a number of effects, such as functioning to promote and enhance inflammation, the processes of phagocytosis and ultimate cell death, known as cytolysis, membrane attack complexes, as well as immune clearance. So we're going to talk about what each of these things means in just a moment. But first, let's consider what activates complement cascades. The intent here is to talk about complement 3 specifically and the way that it mediates responses to help support the functions that we've just discussed. There are 3 different ways that lead to complement 3 activation. The first is known as the classical pathway, and the classical pathway is a system of antibody antigen combination. So this comes from a reaction thanks to our third line of defense. But when antibody antigens are activated, this can promote activity of complement 3 in order to start this cascade of functions. Now, it was known as the classical pathway because this was the first pathway that was discovered for complement 3. However, some additional ways to kick off the complement cascade have also been discovered. The next one we're going to visit about is known as the alternative pathway. It was discovered that while complement assists the third line of defense, it turns out a third line of defense doesn't have to be activated in order for complement to become activated. So the next pathway is known as the alternative pathway. This is a separate mechanism that is going to activate this complement 3 molecule to begin to do the functions that we mentioned earlier. Lastly, it was later discovered that peptidoglycan, as a component of the cell wall located in many microorganisms that are bacterial in nature, can also promote a lectin-based pathway. And so lectin is the third mechanism that has been discovered to kick off this complement cascade activating C3. So let's take a moment to break down the processes that are occurring with complement 3 once it is activated and what kind of effects it has to be able to promote the functions of inflammation, phagocytosis, cytolysis, membrane attack complexes known as MACs, and also immune clearance. Allow me to introduce you to complement 3. Now, complement is named based on the order in which these proteins were discovered. So they are named complement 1 through complement 30, C1, C2, C3, etc. And the focus of this video will be to look at complement 3 as it begins this fascinating cascade that further supports the second line of defense and the actions that are necessary to try to remove a microbe that is penetrated into the body. So we're going to introduce complement 3 here, and you're going to notice on your screen that complement is made of 2 components. We're going to label them A and B. We're going to specifically refer to them as complement 3A and complement 3B. But prior to activation, this is an inactive molecule that is attached to each other, cumulatively known as complement 3. And whether the classical, alternative, or lectin pathway activate this system, when complement 3 gets activated, a very unique thing happens. Boom. When complement 3 gets activated, it's going to go from an immature, large protein structure and crack in half. And as it cracks in half, it's going to make 2 different components, complement C3A and complement C3B. Each of these is going to serve unique functions in the body to help coordinate different lines of defense, different features of the human body in an attempt to promote and really support the immune system in eliminating microbes that have tried to get into the body. So while this is showing us our protein up here, as we move into the next part of the diagram, we're going to zoom in. I'm going to shrink these molecules to suggest that these are the molecules that are signaling molecules that are going to help coordinate activity. And so you're going to see the color used and the shape used, but you're going to notice they're going to become quite a bit smaller. So let's start with considering the actions of complement C3A. What does this molecule do as a signaler once it has been activated and separated from C3B? So, one of the cells that we find in the second line of defense that are going to help promote healing are cells that release histamine. Histamine is yet another signaling compound commonly used to coordinate activity within the human body, specifically responses associated with inflammation. So when C3A is activated and released into circulation, there are receptors that are on the cells that we know produce histamine. So you'll notice mast cells, which are definitely the most common form of histamine releasing cell, but also basophils are listed here. Both of these cells are going to come from similar stem cell lines and have the ability to transcription and translation produce histamine as a signaling molecule. Now, complement 3C3A is going to be able to bind to both mast cells as well as the basophils. When complement C3A binds to a mast cell or binds to a basophil, this acts as a signal and encourages these cells to release their histamine. So histamine is going to be released as an effect, and histamine specifically causes the end result known as inflammation. This is going to promote all of the things that happen during inflammation. And if you are curious about what sort of events happen when inflammation is released, I suggest watching the inflammation video that runs through the signaling processes and how histamine causes vasodilation, causes increase in capillary permeability, and increases the sensation of pain. So one key effect of complement C3A is to signal to the histamine releasing cells that this is a good time to release histamine. Now, in homage to our white blood cells, we have some neutrophils, we have some monocytes, which are going to mature into some macrophages, and the macrophages are illustrated gargantuan because the macrophages are the large cell that goes around phagocytosing things that need to be eliminated from the body. Here's another cool feature. When C3A binds to these cells, that acts as yet again, a signaling molecule. We're going to need more macrophages. We're going to need more neutrophils. And oh, by the way, could you please start to digest things? So when complement C3A binds to the phagocytic cells, this increases, the net result is an increase in phagocytosis. So key role of complement C3A is to support most of those white blood cells, most of those cells and components of the second line of defense as an attempt to encourage the removal of microbes from the area. Now, we also have this other signaling molecule that was produced that's known as complement C3B. So now it's time to start looking at what does complement C3B have the ability to do. In one example, complement C3B is going to act as a signaling molecule, and it's going to help facilitate red blood cells picking up antibody-antigen complexes. So what you'll notice is a little red spidery shaped cell here with some antibodies that are attached to it. This is how complement is helping the third line of defense. So if antibodies exist, they can bind to pathogens, but we then need a way to get those pathogens out of the body. So the body can use complement 3, and complement 3 is going to help adhere this complex of microbe. We'll make it a little bigger so it looks like it's grabbing it. And it's going to help hold and adhere and signal this red blood cell to carry this antibody-antigen complex out of the system. Now, this is a unique idea, and most people are unfamiliar with the fact that red blood cells can help collect things and move them around the body. Where is the red blood cell going with this? Well, red blood cells go through a structure that I commonly think of as the elephant graveyard of your body. It's essentially the white blood cell, red blood cell graveyard of your body. And that structure is called the spleen. And when red blood cells go through the spleen is going to use a series of macrophages, and the macrophages have already been told by C3a that they need to increase their activity. So these red blood cells carry these antibody-antigen complexes to the spleen, where macrophages in the spleen kind of spit and polish. They buff up the red blood cell, essentially removing the complement 3 and removing that antibody-antigen complex. That causes an end result. So we had inflammation and phagocytosis. The end result of this is a system called immune clearance. The word clearance is frequently used in medicine to reference the removal or the clearing of a molecule or a substance from either the bloodstream or from the kidneys as well. So the first action that we're seeing from complement C3b is to coordinate red blood cells carrying around antibody-antigen complexes, carrying those complexes to the spleen, where macrophages, who were already signaled by C3a to increase their activity, will be on site to remove those antibody-antigen complexes and clean the red blood cell off. That is a process known as immune clearance. What if there are no antibodies or antigen complexes? Perhaps this is the first exposure of the body to this foreign cell, and perhaps there are no antibodies present. So the immune clearance function is going to be useful when the third line of defense has already created antibodies, but there are many situations in which the body does not already have antibodies present. Never fear, complement C3b is here. So complement C3b can also bind to and attach to a pathogen, even if there is no antibody complex to it. This is going to help make it more obvious and noticeable to body cells that would have the ability to destroy this foreign invader. This is a process known as opsonization. Opsonization is the process by which the body adds either an antibody, or in this case complement C3b, to help make a pathogen noticeable and help indicate to the cells that can kill that this is something that needs to be removed from the body. So opsonization is a marking for destruction or a mark for removal. And when opsonization takes place, this ideally leads to the death of the cell that was bound by complement C3b. We call this the effect of opsonization. But complement C3b is not done. Not only does it promote immune clearance, not only does it support cytolysis through the action of opsonization, but there are other complement molecules that can further help attack a membrane of a pathogenic cell. So you'll see now noted a series of railroad spike looking structures. They are C5, C6, C7, C8, and C9. These, when activated, have the ability to be driven into a plasma membrane, and they drive themselves in a circular fashion. So essentially what happens here is that complement C3b binds to complement C5. And when complement C3b binds to complement 5, that's the signal that it's time to activate complement 5 through 9. So in a plasma membrane of a cell, okay, we're going to kind of look at this from the top of the cell now. Okay, I have my plasma membrane of my cell. In fact, maybe we should even make it red, right, to go along with this red antigen we've been discovering or fighting. So here's the plasma membrane of the cell. And this is the inside of the cell. And the membrane is keeping the inside in, and here is the outside of the cell with the goal of keeping the outside out and the inside in. When complement C3b attaches to complement 5. It activates complement 5, and complement 5 spears itself through the membrane into this cell. That triggers complement 6 to join, which triggers complement 7 to join, which triggers complement 8 to join, which triggers complement 9 to join. So what we get is this little rosette, right? If we were looking down from the top, we would see C3-5 next to C3-6, next to, I keep saying C3, C7, next to C8, next to C9. And what we get is this little rosette. And what that means is that right here in the center, what was plasma membrane and was adhered to the rest of the plasma membrane of the cell that is being fought against, but now there's nothing holding that little bit of plasma membrane in, so it's going to pop out. Out it goes. And that's going to create a hole. That hole is important then because what was inside now can leak outside, and we've permanently disrupted the membrane. This combined action of complement 5 through 9, creating this little rosette structure, poking holes in cells, is cumulatively referred to as a membrane attack complex. Membrane attack complex. And the abbreviation for that is a MAC, M-A-C. When that membrane attack complex is created, then a really handy thing happens, which is that the semi-permeable structure of the membrane becomes compromised. And as it becomes compromised, that provides an opportunity for the internal cell contents to start to leak out. This is also going to lead to cell death. This is going to end in cytolysis. Time to summarize complement cascade system. So, we began this video with a discussion of 3 different ways that can activate this inactive form of a protein known as complement 3. We discussed the classical pathways using antibody-antigen complexes to cause and activate complement 3. We also looked at other pathways known as the alternative and lectin pathways associated with peptidoglycan as an attempt to activate complement 3. Complement 3 is a large protein. When broken in half, each half has signaling abilities. Complement C3A specifically is going to target mostly the white blood cells, most of the second line of defense. So mast cells will be bound by complement C3A. That promotes the release of histamine and supports the inflammatory process. Simultaneously, complement C3A is also binding to white blood cells that are phagocytic in nature. This includes neutrophils, monocytes, and very importantly, macrophages, which are going to begin to navigate around the body and start to eat more. They're going to do more phagocytosis because C3A is signaling to them they need to increase their activity. Simultaneously, the other half of the inactive complement 3, the piece known as complement C3b, is going to do some additional signaling. One potential signaling mechanism of C3b involves helping connect red blood cells to pathogens that are bound by antibody-antigen complexes. Those red blood cells are then sent to the spleen, which happens to be rich in macrophages, who were told by complement C3a, hey, time to eat more and do more phagocytosis. Those macrophages are going to remove the antibody-antigen complexes located on the red blood cells and cause an effect known as immune clearance, to clear the microbe from the blood. This is only going to be active, though, if we have a third line of defense and antibodies are present. Even without antibodies present, complement C3b also can act like an antibody to help mark a cell for removal. Complement C3b can adhere to foreign cells and opsonize them or mark them for destruction. This helps attract some of those phagocytic cells that were activated by C3a, and they're going to eat and destroy the foreign pathogen in a process known as cytolysis. Additionally, there are other complement proteins, complement levels 1 through 30, And complement C3b has the ability to bind to complement 5 and starts a reaction cascade. When complement 5 is activated, it's going to penetrate the plasma membrane of the cell that needs to die. Complement 5 calls in its buddy complement 6 calls in complement 7, and so forth until we have C5 through C9 stabbed through a plasma membrane to create a ring or membrane attack complex. That disrupts the plasma membrane and allows a bit of the plasma membrane to pop out, thus reducing that semi-permeable membrane function that is so critical for all cells. Now the inside of the cell is going to begin to leak outside of the cell, and this is another feature that lends itself towards cytolysis. So complement may seem scary or initially kind of intimidating, but if we break this down and really think of it as just another signaling molecule to help coordinate the second line of defense, then we can see through this diagram how helpful it is to have complement is a mechanism to be able to really enhance the second line of defense and help protect the human body. I hope this video helped you connect the ideas of how complement can be acting as a signaling molecule. Have a great day.